Hello, and thank you for being here. Tonight, we will slowly drift into a state of calmness and explore the quiet, ancient intelligence of plants, beings that learned to shape this planet long before animals ever walked upon it. As you settle in, imagine a vast green world breathing softly, leaves unfurling toward light, roots weaving patiently through dark living soil. Plants sense, respond, remember, and adapt, using subtle chemical whispers and underground fungal pathways that connect entire forests into shared communities. Through photosynthesis, they transform sunlight into life itself, balancing air, climate, and nourishment with effortless precision. As you listen, you may picture towering trees exchanging nutrients through mycorrhizal networks, desert plants conserving water with remarkable ingenuity, and delicate mosses thriving quietly where little else can. This is not a story of stillness, but of constant, intelligent motion. Slow, rhythmic, and deeply aware. If you enjoy these relaxing moments, please consider leaving a like, subscribing, or sharing your thoughts below. Your support is deeply appreciated and helps others find their way to this peaceful space. You can also turn on the notification so you don't miss any of our future journeys together. But for now, just breathe. Let your body relax, release any tension, and allow your mind to drift. Let's begin. Plants perceive their surroundings through an array of sensory systems that rival those of animals, though expressed in entirely different forms. Roots detect moisture gradients, mineral concentrations, gravity, and physical obstacles, constantly adjusting their growth direction as conditions shift. Leaves register light intensity, wavelength, and duration, allowing plants to measure seasonal change with extraordinary accuracy. Some species respond differently to red and blue light, altering their internal chemistry accordingly. This sensory awareness forms the foundation of plant intelligence, not as conscious thought, but as distributed decision-making woven into every cell. This awareness extends into time itself, as plants possess internal biological clocks that regulate daily and seasonal behavior. Circadian rhythms govern when leaves open, when flowers release pollen, and when defensive compounds are produced. These rhythms persist even in constant darkness, demonstrating that plants maintain an internal sense of time independent of external cues. Seasonal timing allows plants to anticipate droughts, frosts, and insect life cycles, rather than merely reacting to them. In this way, plant intelligence often operates predictively, shaping survival through preparation rather than urgency. Beneath the soil, roots engage in constant information gathering, sampling their environment through chemical receptors along their tips. When a root encounters nutrient-rich soil, it branches aggressively, while poor conditions cause restrained growth elsewhere. This selective expansion resembles strategic investment, allocating energy where returns are highest. Experiments have shown that roots can distinguish between nutrients provided by their own leaves and those belonging to neighboring plants, adjusting behavior based on competition. Such discrimination reveals an ability to process context, not just stimulus, within underground decision systems. Plant memory exists not as stored images, but as biochemical changes that influence future behavior. After experiencing drought, some plants respond more efficiently when water scarcity returns, altering gene expression to conserve resources earlier than before. This form of learning, known as stress priming, can persist across seasons. In certain cases, these adjustments are passed to offspring through epigenetic markers, allowing future generations to inherit environmental knowledge. Memory, in plants, becomes a legacy written into molecular structure rather than neural pathways. Communication between plants occurs through airborne chemicals released by leaves under attack. When insects begin feeding, damaged plants emit specific volatile compounds that drift through the air, alerting nearby plants to prepare defenses. Neighbors respond by producing bitter chemicals, thickening cell walls, or attracting predators that feed on the insects. This chemical messaging system operates across species boundaries and can influence entire ecosystems. Rather than isolated organisms, plants function as participants in a shared atmospheric conversation shaped by threat and response. Below ground, communication becomes even more intricate through mycorrhizal fungal networks connecting plant roots. These fungi form symbiotic partnerships, trading minerals and water for sugars produced through photosynthesis. Within these networks, chemical signals travel from one plant to another, conveying information about stress, disease, or nutrient availability. Researchers have observed carbon transfer from healthy plants to shaded or damaged neighbors, suggesting cooperative behavior. The forest floor thus hosts a living web of exchange that challenges the idea of plants as solitary life forms. Plants are capable of assessing competition and adjusting their growth accordingly. When growing alongside relatives, some species reduce aggressive root expansion, conserving resources for shared survival. In contrast, when surrounded by unrelated plants, roots grow more extensively, competing for space and nutrients. This ability to distinguish kin from non-kin relies on chemical cues in the soil, functioning as a form of recognition. Such behavior implies an evolved strategy, balancing cooperation and competition, shaped by long-term survival rather than immediate dominance. Even without neurons, plants transmit electrical signals through their tissues. Changes in membrane voltage travel along stems and leaves in response to injury, temperature shifts, or sudden darkness. These electrical impulses trigger rapid physiological responses, such as closing pores or activating chemical defenses. In carnivorous plants, electrical signaling coordinates trap movement and digestion. While slower than animal nervous systems, these signals form an internal communication network that allows plants to integrate information across distant tissues. Plants exhibit problem-solving behavior when navigating complex environments. Climbing vines adjust their growth paths when encountering obstacles, redirecting toward available supports. Experiments involving mazes have shown roots preferentially growing towards sound vibrations associated with flowing water. This response occurs even when moisture is absent, suggesting anticipation rather than reaction. Such behaviors demonstrate flexible strategies rather than rigid programming, revealing intelligence expressed through growth patterns rather than movement. The perception of sound in plants has gained scientific support in recent decades. Certain flowers produce sweeter nectar when exposed to the wing vibrations of pollinating insects. Roots grow toward low-frequency vibrations associated with water movement. These responses suggest that plants detect mechanical oscillations and translate them into biochemical action. Sound perception allows plants to gather information beyond light and chemistry, expanding the sensory world available to stationary organisms rooted in place. Plants defend themselves, using a diverse arsenal of chemical compounds that rival pharmaceutical laboratories. Alkaloids, tannins, and terpenes deter herbivores, disrupt digestion, or interfere with insect nervous systems. The production of these chemicals is carefully regulated, increasing during periods of attack and decreasing when threats subside. Some compounds are synthesized only after specific signals are received, conserving energy. This dynamic defense strategy reflects decision-making based on cost, risk, and environmental context. Certain plants manipulate the behavior of insects with remarkable precision. Some emit chemicals that mimic insect pheromones, luring predators of herbivores to the scene. Others alter the development of insects feeding on them, slowing growth or reducing reproduction. These interactions reflect a deep evolutionary history of behavioral influence without movement. Plants shape animal actions through chemistry, effectively outsourcing mobility to other organisms while remaining rooted in place. Light perception in plants extends far beyond brightness. Specialized photoreceptors detect ultraviolet, blue, red, and far-red wavelengths, allowing plants to sense shading by neighbors. When light quality changes, plants adjust stem length, leaf orientation, and flowering time. This ability to read spectral information enables plants to anticipate competition before it occurs. Rather than reacting to physical contact, they respond to subtle shifts in light composition that signal nearby growth. Plants manage energy with extraordinary efficiency, constantly balancing growth, defense, and reproduction. When resources are scarce, growth slows while maintenance systems remain active. During abundance, energy is redirected toward expansion or seed production. These trade-offs are regulated by complex hormonal networks, integrating multiple signals simultaneously. The result is a continuous assessment of internal and external conditions, resembling an economic system rather than a simple stimulus-response mechanism. Some plants can count. The Venus flytrap, for example, measures the number of times its trigger hairs are stimulated before closing. This counting determines how tightly the trap seals and how many digestive enzymes are released. The plant adjusts its response based on prey size and struggle intensity. This numerical assessment ensures energy is not wasted on false alarms, demonstrating that even brief electrical events can be integrated into meaningful decisions. Plants also respond to touch in nuanced ways. Repeated physical contact can cause stems to thicken, becoming more resistant to wind or grazing. This phenomenon, known as thigmomorphogenesis, alters gene expression and structural development. The response depends on frequency and intensity, not merely presence. Through touch perception, plants adapt their architecture to environmental stress, reinforcing structures where needed and conserving material elsewhere. Root systems map their surroundings in 3 dimensions, avoiding obstacles while exploiting open soil. When encountering stones or compacted earth, roots change direction, branching or flattening as needed. This spatial awareness emerges from localized sensing rather than centralized control. Each root tip processes its immediate environment, collectively producing coherent architecture. Intelligence here is distributed, arising from many small decisions rather than a single command center. Plants interact with microbes in ways that resemble selective partnerships. Roots release sugars and signaling molecules that attract beneficial bacteria while discouraging harmful ones. These microbial communities enhance nutrient uptake, suppress disease, and influence growth patterns. Plants actively shape their microbiome, adjusting chemical output based on need. This relationship expands the plant's functional capabilities, effectively outsourcing certain tasks to microscopic allies. Stress signals in plants can travel long distances internally, allowing undamaged tissues to prepare defenses before threats arrive. When one leaf is attacked, signals propagate through vascular tissue, activating protective responses elsewhere. This internal alert system operates through hormones, electrical changes, and reactive molecules. Such coordination ensures that the entire organism responds as a unified system, despite lacking centralized awareness. Over evolutionary time, plant intelligence has been shaped by immobility. Unable to flee, plants evolved sensitivity, prediction, and chemical influence as primary tools for survival. Every leaf and root participates in information processing, creating a slow but resilient form of cognition. This intelligence unfolds over hours, days, and seasons, embedded in growth and structure. In understanding plants, we encounter a form of thinking fundamentally different from our own, yet no less intricate. Plants interpret gravity with remarkable precision, allowing roots to descend and shoots to rise regardless of orientation. Specialized cells called statocytes contain dense, starch-filled structures that settle downward, informing growth direction. When a plant is rotated, these particles shift position, triggering hormonal redistribution that redirects growth. This gravitational awareness is not instantaneous, but measured, unfolding gradually as signals propagate through tissues. By continuously reassessing orientation, plants maintain spatial coherence even after storms, landslides, or animal disturbance. Gravity perception forms a foundational reference point, anchoring all other environmental decisions within a stable internal map. Hormones serve as the chemical language coordinating plant behavior across distant tissues. Auxins regulate growth direction, cytokinins influence cell division, gibberellins control elongation, and jasminates activate defense. These compounds rarely act alone. Instead, they interact in shifting ratios that fine-tune responses. A single stimulus can produce different outcomes depending on hormonal balance, developmental stage, and environmental context. This layered signaling allows nuanced regulation rather than binary reactions. Plant intelligence emerges from these overlapping chemical conversations, producing outcomes that appear intentional despite lacking centralized oversight. Plants assess risk before deploying costly defenses. Producing toxins or structural barriers requires significant energy. So many species wait for reliable confirmation of threat. Some insects leave characteristic saliva compounds while feeding, which plants recognize as authentic danger signals. Only then do defensive pathways fully activate. This discrimination prevents unnecessary expenditure triggered by harmless contact like rain or debris. Such risk evaluation mirrors cost-benefit analysis, where the plant weighs probability against consequence before committing resources to protection. Seed germination reflects sophisticated environmental assessment rather than automatic growth. Seeds measure temperature fluctuations, moisture levels, oxygen availability, and light exposure before breaking dormancy. Some species require specific seasonal cues, ensuring seedlings emerge during favorable conditions. Others delay germination for years, maintaining viability until circumstances improve. This patience prevents population collapse during unfavorable periods. Within each seed lies a decision framework shaped by evolutionary history, allowing future plants to synchronize emergence with ecological opportunity. Plants also influence soil chemistry to improve survival prospects. Through root exudates, they release organic acids, enzymes, and sugars that alter nutrient availability. These substances can mobilize phosphorus, neutralize toxins, or reshape microbial populations. By actively modifying their immediate environment, plants extend intelligence beyond physical boundaries. Soil becomes a responsive medium rather than passive substrate. This capacity to engineer surroundings allows plants to thrive in conditions that would otherwise limit growth. Some plant species monitor neighbor density using chemical cues exuded by roots. As population increases, these signals intensify, triggering reduced growth rates or altered architecture. This self-thinning behavior prevents overcrowding and resource depletion. Rather than competing until collapse, plants adjust collectively through chemical feedback. Such regulation stabilizes populations over time, demonstrating intelligence expressed at community scale rather than individual ambition. Plants detect atmospheric changes with surprising sensitivity. Shifts in carbon dioxide concentration influence stomatal behavior, altering gas exchange and water retention. Rising ozone levels trigger antioxidant production to prevent cellular damage. Even subtle changes in humidity affect transpiration rates and leaf orientation. This constant monitoring allows plants to balance photosynthesis with water conservation. Atmospheric awareness ensures survival in fluctuating climates, integrating invisible signals into daily physiological decisions. The ability to remember seasonal cycles allows perennial plants to coordinate long-term strategies. Dormancy, bud formation, and flowering schedules rely on cumulative exposure to cold or warmth rather than isolated events. This prevents premature responses to brief weather anomalies. Plants effectively average environmental data over time, ensuring stability. Such temporal integration reflects a sophisticated understanding of pattern versus noise, allowing long-lived species to persist across decades. Some plants manipulate soil temperature using leaf litter and canopy structure. By shading ground surfaces or trapping insulating layers, they influence microbial activity and nutrient cycling. These modifications affect root function and seasonal growth timing. Plants thereby shape microclimates to align with their physiological needs. Environmental control becomes an extension of intelligence, subtly steering physical conditions without movement. Plants respond differently to damage depending on location. Injury to reproductive structures triggers stronger defensive responses than damage to older leaves. This prioritization reflects an internal valuation system, protecting tissues most critical for future survival. Signals from damaged areas carry contextual information, not just alerts. The plant allocates resources accordingly, reinforcing strategic importance within its own body plan. Some species use mimicry to reduce predation. Certain plants resemble toxic neighbors or produce leaf shapes that deter feeding. Others alter coloration to resemble diseased or nutrient-poor specimens, discouraging herbivores. These visual strategies require perception of predator behavior over evolutionary time. While individual plants do not choose appearance, intelligence emerges through inherited design shaped by selective pressure and environmental feedback. Plants adjust water usage dynamically through stomatal regulation. Opening and closing these microscopic pores balances carbon intake against water loss. Signals from roots detecting drought can influence leaves meters above ground. This coordination ensures survival during extended dry periods. The plant integrates information from soil, air, and internal reserves to manage hydration strategically rather than reflexively. Electrical signaling in plants can propagate at different speeds depending on stimulus intensity. Stronger disturbances generate faster or broader signals, activating more extensive responses. This graded communication allows proportional reactions rather than uniform output. The system resembles analog processing, where magnitude influences outcome. Plants thus encode information not just in presence, but in degree. Plants also demonstrate anticipation through developmental timing. Some species accelerate flowering when sensing competition, prioritizing reproduction before resources vanish. Others delay growth until competitors fade. These adjustments rely on detecting chemical markers from neighbors. Anticipation allows plants to act ahead of direct conflict, favoring foresight over confrontation. Root systems avoid toxic zones using chemical detection. Heavy metals, salt concentrations, or harmful microbes trigger directional changes. Roots curve away from danger while exploring safer regions. This avoidance behavior reduces long-term damage and improves nutrient acquisition. Spatial intelligence arises from continuous assessment and redirection, building a subterranean map of opportunity and risk. Plants interact with fire regimes in intelligent ways shaped by evolution. Some species require heat or smoke chemicals to germinate. Others store seeds in fire-resistant cones that open only after exposure. These adaptations synchronize regeneration with post-fire conditions when competition is reduced and nutrients abundant. Fire becomes a signal rather than catastrophe within certain ecosystems. Some plants alter flowering time in response to pollinator availability. Changes in temperature or insect populations influence bloom schedules. This synchronization maximizes reproductive success while conserving energy. Timing becomes a responsive dialogue between plant and ecosystem. Rather than fixed calendars, plants follow living cues shaped by mutual dependence. Plants measure internal energy reserves before committing to reproduction. Flowering and seed production demand extensive resources. When reserves are insufficient, plants delay or reduce output. This restraint ensures survival beyond a single season. Internal accounting systems balance present opportunity against future viability. The intelligence of plants challenges human definitions centered on speed and cognition. The decision-making unfolds across growth, chemistry, and time. Each response reflects accumulated information processed without awareness as humans understand it. Intelligence here is not diminished by slowness, but refined through persistence. Plants reveal that thinking does not require minds, only systems capable of learning, adapting, and enduring. Plants can distinguish between different types of damage and respond with tailored strategies. A leaf torn by wind triggers minimal response, while feeding by insects activates complex chemical cascades. This distinction arises from molecular patterns detected in insect saliva, which differ from mechanical injury. Upon recognition, plants initiate localized and systemic defenses, altering metabolism across the organism. Such specificity prevents unnecessary energy loss and reflects a refined interpretive capacity. Rather than reacting blindly, plants analyze cause and context, revealing intelligence rooted in discrimination rather than speed. Some plants possess the ability to suppress growth temporarily as a survival tactic. During prolonged stress, such as drought or nutrient scarcity, growth-related genes are downregulated while maintenance pathways remain active. This suspension preserves structural integrity until favorable conditions return. When resources improve, growth resumes with surprising efficiency. This reversible slowdown mirrors strategic retreat rather than failure. Plants thereby avoid irreversible damage, demonstrating foresight embedded in molecular regulation rather than conscious planning. Plants coordinate development across vast internal distances using vascular signaling. Hormones and electrical impulses travel through xylem and phloem, integrating information from roots, stems, and leaves. A nutrient deficiency detected underground can influence leaf architecture above ground. This long-range coordination ensures the organism responds as a unified whole rather than isolated parts. Intelligence emerges from integration, where local information contributes to global outcomes. The plant body functions as a communication network, shaped by flow rather than hierarchy. Some plants modify leaf orientation throughout the day to regulate temperature and light exposure. This movement reduces overheating during intense sunlight and maximizes photosynthesis during optimal conditions. These adjustments are driven by changes in water pressure within specialized cells, responding to environmental signals. By modulating exposure, plants protect delicate tissues while sustaining energy production. Such regulation reflects continuous monitoring and adjustment, akin to real-time environmental management. Plants also exhibit selective investment in offspring. When resources are limited, some species produce fewer seeds but enhance their size and nutrient content. Larger seeds offer higher survival probability in challenging environments. Conversely, abundant conditions favor quantity over individual investment. This flexibility reflects an internal assessment of environmental quality, balancing risk and reward across generations. Reproductive strategy becomes a calculated response rather than fixed behavior. Roots communicate internally using chemical gradients that guide branching patterns. Areas with higher nutrient concentration stimulate lateral root formation, while pore zones suppress expansion. This spatial regulation prevents wasted growth and improves efficiency. Each root segment contributes localized data, collectively shaping overall architecture. Intelligence arises through accumulation of small decisions, resulting in coherent, adaptive structures beneath the surface. Plants respond to repeated stress with altered sensitivity. After multiple exposures to similar threats, responses become faster and more robust. This phenomenon reflects molecular priming, where prior experience reshapes future reactions. Unlike reflexive systems, plants adjust thresholds dynamically. Experience modifies behavior, fulfilling a key criterion of intelligence. Learning in this context is biochemical rather than cognitive, yet its effects are enduring and measurable. Some plants manipulate nutrient flows to favor certain tissues during stress. When resources decline, growth shifts toward roots rather than leaves, improving water and mineral uptake. This internal reallocation enhances survival probability. The plant evaluates which structures offer the greatest return under current conditions. Strategic redistribution mirrors decision-making, grounded in internal priorities rather than external compulsion. Plants interact with seasonal pathogens through anticipatory defense. As conditions favor fungal growth, Plants increase production of antifungal compounds preemptively. This timing reduces infection severity before visible symptoms appear. Anticipation relies on recognizing environmental patterns rather than immediate threat. Such preparedness underscores intelligence shaped by long-term ecological rhythms. Plants detect chemical signatures left by previous occupants in soil. These signals influence germination success, growth rate, and disease resistance. Some plants avoid soils previously occupied by competitors, while others exploit beneficial residues. This awareness of ecological history allows informed placement within the landscape. Soil memory becomes an extension of plant perception. Plants adjust internal water transport pathways in response to prolonged dryness. Xylem structure changes to reduce vulnerability to air bubbles that disrupt flow. These anatomical modifications improve resilience during future droughts. Structural adaptation based on past conditions reflects long-term learning expressed physically. The plant body becomes a record of environmental experience. Some plants engage in deceptive signaling to avoid herbivory. By emitting stress-related chemicals even when unharmed, they create false alarms that deter feeding. This strategy exploits predator behavior shaped by evolutionary learning. Deception emerges without intent, yet functions effectively within ecological systems. Intelligence here operates through influence rather than awareness. Plants regulate nutrient uptake using feedback mechanisms that prevent toxicity. When internal levels rise too high, absorption slows or halts. Excess minerals can damage tissues, so balance is essential. Continuous monitoring ensures stability. Homeostasis represents intelligence devoted to equilibrium rather than growth alone. Plants exposed to irregular light patterns adjust photosynthetic machinery accordingly. Shaded environments trigger increased chlorophyll concentration, improving efficiency under low light. Sudden brightness initiates protective responses to prevent cellular damage. This adaptability maintains energy production across variable conditions. Intelligence manifests as optimization under constraint. Some species coordinate flowering across populations using environmental cues. Synchronization increases pollination success and genetic exchange. This coordination arises from shared sensitivity rather than communication. Collective timing transforms individual behavior into population-level intelligence shaped by environmental alignment. Plants respond to salt stress by altering cellular ion balance. Specialized transporters remove excess sodium while retaining essential potassium. This regulation preserves enzyme function and cellular stability. Adaptive ion management reflects detailed internal monitoring. Survival in harsh soils depends on this biochemical intelligence. Plants exposed to grazing pressure develop tougher tissues over time. Increased fiber content reduces palatability and damage. This structural reinforcement depends on repeated exposure, illustrating experience-dependent change. Adaptation becomes embedded in form rather than behavior. Some plants influence water flow in soil by altering root density. Dense root zones slow runoff, increasing absorption. Sparse areas allow drainage. By shaping hydrology, plants modify resource availability. Environmental engineering becomes an extension of intelligence. Plants coordinate senescence, the aging process, to recycle nutrients efficiently. As leaves decline, valuable compounds are relocated to storage tissues. This controlled dismantling conserves resources for future growth. Senescence reflects deliberate redistribution rather than decay. Plant intelligence challenges assumptions about cognition itself. Without thoughts or awareness, plants sense, integrate, predict, and adapt. Their intelligence is architectural, chemical, and temporal. Understanding it expands the concept of thinking beyond minds, revealing cognition as a property of living systems shaped by interaction and time. Plants monitor internal nutrient ratios with remarkable precision, adjusting uptake and storage to avoid imbalance. Nitrogen, phosphorus, potassium, and trace minerals must remain within specific ranges for enzymes to function correctly. When one element becomes scarce, plants alter root behavior and transporter activity to compensate. Excess levels trigger sequestration or reduced absorption. This internal accounting system resembles regulation in engineered networks, where stability depends on constant feedback. Intelligence emerges through balance, allowing plants to thrive in soils that fluctuate unpredictably over time. Some plants exhibit directional growth toward chemical signals released by beneficial organisms. Roots grow towards symbiotic fungi and bacteria that enhance nutrient acquisition. This targeted expansion reflects selective responsiveness rather than random exploration. Chemical gradients act as invisible guides, shaping subterranean architecture. By choosing allies through growth patterns, plants improve resilience and efficiency. The soil becomes a landscape of signals interpreted continuously, reinforcing the idea that plant intelligence extends beyond the organism into its surrounding environment. Plants adapt leaf thickness and surface texture in response to long-term climate conditions. In arid regions, thicker leaves reduce water loss, while humid environments favor thinner structures for efficient gas exchange. These changes occur gradually, guided by cumulative exposure rather than single events. Structural intelligence emerges over developmental time, embedding environmental knowledge directly into form. Each leaf becomes a record of conditions experienced, translating climate into anatomy. Plants sense mechanical stress from wind and adjust growth accordingly. Repeated bending triggers thicker stems and altered fiber orientation, increasing resistance. This response prevents structural failure without excessive resource investment. The plant measures frequency and intensity, not just occurrence. Mechanical awareness ensures survival in exposed landscapes, where flexibility and strength must be carefully balanced. Intelligence here is expressed through material science rather than behavior. Some plants regulate pigment production to protect against excess radiation. Increased sunlight triggers synthesis of protective compounds that absorb or reflect harmful wavelengths. These pigments reduce cellular damage while maintaining photosynthetic capacity. Regulation depends on light quality, duration, and intensity. This nuanced response allows plants to inhabit environments with extreme exposure. Protective coloration becomes an adaptive interface between organism and atmosphere. Plants detect oxygen availability in soil and modify root metabolism accordingly. Waterlogged conditions reduce oxygen, prompting shifts toward anaerobic processes. Root growth slows, and air-filled tissues may form to facilitate gas exchange. These adjustments prevent energy collapse under suffocating conditions. Oxygen sensing ensures survival in wetlands, floodplains, and compacted soils. Intelligence here operates the boundary between chemistry and survival. Some plants influence pollinator behavior through timed chemical release. Floral scents intensify during peak pollinator activity and fade outside those windows. This synchronization conserves resources while maximizing reproductive success. Timing depends on internal clocks and environmental cues. The plant aligns its signaling with external rhythms, creating efficient partnerships without movement. Intelligence emerges through temporal coordination. Plants respond to ultraviolet exposure by repairing DNA damage using specialized enzymes. Detection of molecular injury triggers repair pathways that preserve genetic integrity. These systems operate continuously, correcting errors caused by radiation or replication. Maintenance intelligence ensures long-term survival across generations. Without such vigilance, mutation rates would threaten viability. Repair becomes a silent but essential form of problem-solving. Some plants adjust root depth seasonally. During dry periods, deeper roots access stable moisture, while shallow roots dominate during rains to capture surface nutrients. This dynamic restructuring reflects anticipation of seasonal patterns. The root system remains adaptable, reshaping itself in response to long-term trends. Spatial intelligence unfolds beneath the surface, unseen but decisive. Plants evaluate shade not only by reduced light, but by changes in spectral composition. Far-red light increases under dense vegetation, signaling nearby competitors. In response, plants elongate stems or reposition leaves to access brighter zones. This early detection prevents loss of photosynthetic capacity. Intelligence lies in interpreting subtle signals before competition becomes overwhelming. Plants regulate carbohydrate storage based on future needs. Sugars produced during photosynthesis are allocated to growth, defense, or storage tissues. During favorable periods, reserves accumulate. During stress, stored energy sustains metabolism. This foresight prevents starvation during extended hardship. Internal budgeting mirrors long-term planning without conscious deliberation. Some plants alter root exudates to discourage pathogens when infection risk rises. Chemical output shifts toward antimicrobial compounds, reshaping microbial communities. This preemptive defense reduces disease severity. The plant does not wait for damage, but adjusts conditions proactively. Intelligence here lies in modifying probability rather than reacting to outcomes. Plants respond differently to chronic stress than acute stress. Short-term challenges trigger immediate defenses, while prolonged exposure leads to structural and metabolic reconfiguration. This distinction prevents exhaustion of emergency responses. Adaptation becomes sustainable rather than reactive. The plant recognizes duration as a critical variable, refining its strategy accordingly. Certain plants synchronize seed release with environmental events such as rainfall or temperature shifts. Timing ensures dispersal occurs under conditions favorable for germination. Sensors detect subtle atmospheric changes, guiding release mechanisms. Reproductive success depends on this precision. Intelligence manifests as alignment with natural cycles rather than forceful expansion. Plants monitor internal water pressure to maintain cellular integrity. Changes in pressure influence growth, signaling drought or saturation. Adjustments prevent tissue collapse or rupture. This hydraulic awareness integrates physics into biology. Stability depends on constant recalibration, ensuring survival under fluctuating conditions. Some plants modify canopy architecture to regulate internal light distribution. Leaf angles shift to prevent self-shading while maximizing overall photosynthesis. This optimization depends on plant size, neighbor proximity, and sun angle. Architecture becomes adaptive rather than fixed. Intelligence shapes form through continuous assessment. Plants exposed to herbivory may increase production of compounds that reduce digestibility rather than toxicity. Tougher tissues slow feeding and reduce damage. This strategy minimizes energy expenditure while remaining effective. Defense becomes subtle rather than extreme, reflecting strategic restraint. Plants regulate seed dormancy depth based on environmental unpredictability. In unstable climates, seeds remain dormant longer, spreading risk across years. This bet hedging strategy increases long-term survival. Intelligence extends beyond individuals into population resilience. Plants sense internal aging signals and coordinate resource withdrawal before tissue loss. Nutrients are relocated efficiently, supporting new growth elsewhere. This controlled transition prevents waste. Aging becomes an organized process rather than decline. Plant intelligence reveals itself through systems that sense, integrate, predict, and adjust. Without awareness or intention, plants solve problems through chemistry, structure, and time. Their intelligence is not diminished by silence or slowness. It is refined through endurance, proving that thinking can exist wherever life meets complexity. Plants interpret nutrient shortages as distinct physiological challenges rather than a single state of deprivation. A deficiency in nitrogen alters protein synthesis and leaf coloration, while insufficient iron disrupts chlorophyll. Formation without affecting overall growth rate. Each shortage activates specialized molecular pathways, guiding targeted adjustments rather than broad stress responses. This internal diagnostic capacity prevents inefficient reactions and accelerates recovery when conditions improve. Intelligence here functions as precision, allowing plants to address specific problems with customized biochemical solutions instead of generalized alarms. The pace at which a plant grows is shaped by its expected lifespan and environmental predictability. Short-lived species compress development, allocating resources rapidly toward reproduction before conditions shift. Long-lived plants invest in durable tissues, slower expansion, and storage systems that support survival across decades. These contrasting strategies arise from internal regulatory networks responding to climate, competition, and disturbance frequency. Growth speed becomes a calculated expression of risk management, aligning biological tempo with ecological uncertainty. Plants detect minute vibrations in their surroundings and translate them into preparatory responses. Movements caused by approaching animals, feeding insects, or shifting substrates generate oscillations that travel through air and soil. Certain frequencies trigger defensive chemistry even before contact occurs. This anticipatory mechanism allows plants to respond to threat probability rather than confirmed damage. Intelligence emerges through sensitivity, transforming distant motion into early biochemical readiness. Some plants influence their surroundings by releasing compounds that reshape soil dynamics. These substances can suppress germination of nearby competitors, alter nutrient availability, or shift microbial communities. The effects accumulate gradually, modifying the ecological balance over time. Rather than direct confrontation, plants achieve advantage by adjusting shared conditions. Intelligence operates through environmental engineering, where success depends on persistence and subtle influence rather than force. Photosynthesis is regulated not only by sunlight, but by internal demand for energy. When carbohydrates accumulate beyond immediate needs, Plants reduce photosynthetic activity to prevent metabolic imbalance. This feedback system prioritizes stability over maximum production. Excess energy can disrupt cellular processes, so moderation becomes essential. Intelligence here reflects restraint, ensuring that productivity remains aligned with utilization rather than availability. Plants dynamically adjust investment between below-ground and above-ground structures. When nutrients are scarce but light plentiful, resources flow toward roots. In shaded environments with rich soil, stems and leaves receive priority. This redistribution optimizes overall performance under shifting constraints. The plant continuously evaluates where energy will yield the greatest return, demonstrating intelligence through proportional allocation rather than fixed growth patterns. Repeated exposure to temperature extremes reshapes plant physiology. Cell membranes alter composition to maintain flexibility, while protective proteins accumulate to stabilize enzymes. These changes persist beyond the initial stress, improving tolerance during future events. Such acclimation reflects learning embedded in structure and chemistry rather than genetics. Experience leaves a lasting imprint, refining responses without altering DNA sequence. As plants increase in size, internal crowding becomes a challenge. Growth slows or redirects when tissues grow too dense, preventing inefficiency and mechanical strain. This self-regulation ensures that expansion maintains functionality. Spatial awareness emerges through localized feedback, coordinating form across the organism. Intelligence governs shape by limiting excess, preserving coherence as complexity increases. Some plants stagger fruit development across time instead of synchronizing ripening. This distribution reduces vulnerability to predators, disease, or sudden weather events. By diversifying timing, the plant spreads reproductive risk within a single season. Such variability increases the likelihood that some offspring survive unfavorable conditions. Intelligence appears as strategic uncertainty, avoiding reliance on a single outcome. Chemical composition within leaves varies according to age and position. Newly formed leaves often receive greater defensive investment, while older tissues store nutrients for redistribution. This internal prioritization reflects future-oriented resource management. The plant protects what holds the greatest potential while recycling what has already contributed. Intelligence assigns value internally, guiding resource flow without centralized control. Plants respond rapidly to brief nutrient abundance. When minerals suddenly become available, transport mechanisms intensify and nearby roots expand. This responsiveness allows plants to capitalize on transient opportunities before competitors intervene. Intelligence here depends on readiness, enabling swift adjustment within biological limits. Success favors sensitivity rather than speed alone. Water vapor release from leaves influences surrounding humidity. By adjusting this release, plants can cool tissues, regulate gas exchange, and limit pathogen development. These changes subtly modify the microenvironment immediately surrounding the plant. Intelligence extends outward, shaping conditions rather than merely adapting to them. Environmental modulation becomes a survival strategy. Information flows continuously between roots and shoots. Signals originating underground influence leaf behavior, while light conditions above ground reshape root architecture. This bidirectional communication ensures coherence across the entire organism. Intelligence arises through integration, preventing contradictory responses that would undermine survival. Plants regulate enzyme synthesis based on actual metabolic demand. When substrates accumulate, enzyme production declines to conserve energy. This fine-tuned regulation prevents waste and maintains equilibrium. Intelligence manifests as efficiency, matching capacity to necessity rather than excess. The aging process in plants is flexible rather than fixed. Favorable conditions can delay tissue decline, extending productive lifespan. Hormonal adjustments suppress degradation pathways, allowing continued photosynthesis and growth. Aging becomes responsive rather than inevitable. Intelligence reshapes time within biological constraints. In mineral-rich but toxic soils, plants isolate harmful elements within specialized compartments. By confining danger, they prevent interference with essential processes. This internal segregation enables survival in environments otherwise hostile to life. Intelligence manages risks spatially, transforming hazard into containment. Plants alter pollen production based on environmental reliability. Unpredictable conditions trigger increased output, improving fertilization odds. Stable conditions favor conservation. Reproductive effort adjusts dynamically to expectation rather than tradition. Intelligence balances expenditure against probability. Damage accumulation within tissues activates maintenance systems before visible failure occurs. Repair pathways address oxidative stress and protein damage, preserving long-term function. Preventative action ensures endurance over extended lifespans. Intelligence prioritizes upkeep alongside growth. Cell differentiation depends on positional information rather than fixed destiny. Chemical gradients guide cells toward specific roles based on location and need. Development responds to context, allowing flexible architecture. Intelligence shapes form relationally rather than rigidly. Plant intelligence emerges as a continuous negotiation with reality. Through sensitivity, memory, integration, and adjustment, plants navigate complex environments without awareness or intent. Their cognition unfolds slowly, embedded in structure and chemistry, revealing intelligence as persistence shaped by time. Plants evaluate rainfall not as isolated events, but as cumulative patterns unfolding over weeks and months. Repeated moisture alters root permeability, shifts microbial alliances, and recalibrates internal water transport systems. A single storm may trigger temporary responses, yet only sustained trends provoke structural change. This distinction prevents false adaptation to short-lived conditions. Intelligence here lies in temporal filtering, allowing plants to align survival strategies with genuine climatic rhythms rather than misleading fluctuations. Cellular expansion in plants depends on exquisitely regulated internal pressure. Water pushes against cell walls, driving growth, yet uncontrolled force would rupture tissue. Plants adjust wall composition, elasticity, and enzymatic remodeling to permit gradual enlargement without structural failure. This microscopic governance integrates physics and chemistry into living form. Intelligence operates at the boundary between strength and flexibility, shaping architecture through controlled tension rather than brute expansion. In environments with persistent shade, plants refine internal photosynthetic chemistry rather than merely enlarging leaves. Enzyme ratios shift to extract energy more efficiently from limited light. Chloroplast arrangement changes to improve photon capture. These refinements conserve resources while enhancing performance. Intelligence favors internal optimization over visible growth, demonstrating that improvement does not always require enlargement, only recalibration. Plants prepare for seasonal insect pressure by interpreting environmental signals that precede outbreaks. Rising temperatures, humidity changes, and day-length patterns activate defensive pathways before herbivores appear. This forecasting reduces damage by ensuring readiness rather than reaction. Intelligence emerges through anticipation shaped by evolutionary memory, allowing plants to respond to probability rather than certainty. As plants age and increase in size, internal transport routes undergo continuous revision. Vascular tissues reorganize to prioritize active growth zones and storage regions. Older pathways gradually diminish in importance while new conduits form. This infrastructural remodeling maintains efficiency despite growing complexity. Intelligence manages internal logistics, ensuring resources reach destinations without bottlenecks as the organism expands. Plants living in nutrient-poor habitats often adopt slower but more efficient metabolic strategies. Enzymatic reactions become finely tuned, minimizing waste and maximizing yield from scarce inputs. Growth slows deliberately, favoring endurance over speed. Intelligence here manifests as restraint, thriving through conservation rather than acceleration in environments where excess would prove fatal. Plants monitor amino acid balance internally, adjusting synthesis when specific compounds accumulate excessively. Production slows or redirects to maintain chemical harmony. This regulation prevents metabolic congestion that could impair cellular function. Intelligence operates through equilibrium, maintaining internal order through continuous feedback rather than abrupt intervention. Root hair density changes dynamically in response to soil structure. Compact substrates stimulate dense hair formation, increasing surface contact for absorption. Loose soils require fewer extensions. This targeted adjustment enhances efficiency without unnecessary expenditure. Intelligence matches anatomical detail to physical context, optimizing interaction with the underground environment. Pigment diversity in plants serves communicative purposes beyond protection. Subtle variations influence pollinator behavior and deter certain herbivores. Color becomes a signaling interface shaped by evolutionary negotiation. Intelligence expresses itself visually, guiding relationships through appearance rather than motion. Persistent wind exposure reshapes plant structure from within. Fiber alignment changes to enhance flexibility, allowing tissues to bend without breaking. Strength emerges through resilience rather than rigidity. Intelligence integrates mechanical stress into development, crafting forms suited for continual movement. Plants assess internal carbon reserves before sustaining defensive output. When energy declines, protection is temporarily reduced in favor of basic maintenance. This prioritization prevents self-inflicted depletion. Intelligence weighs survival against confrontation, choosing restraint when resources demand caution. Some plants synchronize root expansion with cycles of microbial abundance. Growth accelerates when beneficial organisms peak, maximizing nutrient exchange. Timing aligns biological rhythms across kingdoms. Intelligence coordinates partnerships through temporal alignment rather than direct communication. Plants exposed to acidic soils adjust ion transport and enzyme stability to maintain metabolic function. These internal shifts neutralize chemical stress without altering external conditions. Intelligence transforms hostile environments into survivable spaces through biochemical adaptation. In regions prone to disturbance, plants favor modular growth strategies. Structures are designed for regeneration rather than permanence. Damage becomes recoverable rather than catastrophic. Intelligence prioritizes resilience, embedding recovery potential into form. Flowering depends on accumulated daylight rather than individual bright days. Plants measure exposure across time to avoid premature reproduction. Intelligence distinguishes meaningful patterns from noise, ensuring developmental decisions reflect true seasonal change. Antioxidant levels rise and fall in response to metabolic intensity. Increased activity generates more reactive byproducts, requiring greater protection. Intelligence scales defense to internal demand, maintaining cellular integrity dynamically. Under stress, plants alter root secretions to recruit specific microbial partners. These allies enhance nutrient access or suppress disease. Intelligence expands functional capacity through chemical invitation rather than self-sufficiency. Seed coat thickness varies according to environmental unpredictability. Harsh conditions favor tougher barriers that delay germination until circumstances improve. Intelligence embeds patience into structure, spreading survival across time. Fluctuations in internal calcium concentrations act as signals guiding stress responses and development. Calcium functions as a universal messenger, coordinating rapid adjustments across tissues. Intelligence relies on chemical languages that integrate information efficiently. Plant intelligence does not pursue awareness or control. It pursues continuity. Through sensing, memory, integration, and adaptation, plants persist across uncertainty. Their cognition unfolds slowly, written into structure and chemistry, revealing intelligence as endurance shaped by time. Plants evaluate temperature not only through absolute values, but through rate of change. A gradual cooling triggers different physiological pathways than a sudden drop, even if the final temperature is identical. This distinction allows plants to prepare tissues incrementally when conditions shift slowly, while rapid changes activate emergency stabilization responses. Enzymes, membranes, and transport systems adjust based on perceived trajectory rather than endpoint. Intelligence here lies in sensitivity to motion within the environment, enabling plants to respond appropriately to trends rather than static measurements. Plants regulate internal signaling noise to prevent overreaction. Many stimuli generate weak or ambiguous signals, which are filtered through threshold systems before triggering full responses. Only when signals surpass specific intensity or duration limits do costly pathways activate. This filtration prevents exhaustion caused by constant low-level disturbance. Intelligence emerges through discernment, ensuring that responses remain proportional and sustainable rather than reactive to every fluctuation. Plants track soil moisture distribution with remarkable spatial precision. Rather than treating the underground environment uniformly, roots detect localized pockets of water and expand selectively toward them. Drier zones are avoided or deprioritized. This selective exploration reduces wasted growth and improves efficiency. Intelligence unfolds as spatial mapping beneath the surface, where roots collectively chart a dynamic landscape of opportunity and constraint. Plants exposed to recurring nutrient pulses adjust uptake timing rather than increasing constant absorption. Transport mechanisms become more responsive during expected availability windows, then relax during scarcity. This rhythmic sensitivity conserves energy while maximizing gain. Intelligence aligns physiology with environmental cadence, synchronizing internal processes to external cycles, rather than maintaining continuous readiness. Plants modify internal sugar transport routes based on demand distribution. Actively growing tissues receive priority, while surplus is diverted to storage organs. When growth slows, stored sugars are redistributed. This dynamic rerouting maintains balance across the organism. Intelligence manages internal logistics, ensuring that energy flows toward necessity rather than habit. Some plants alter stem flexibility based on developmental stage. Young tissues remain supple, allowing adjustment to surroundings, while mature tissues stiffen to provide support. This progression balances adaptability with stability. Intelligence coordinates mechanical properties over time, matching structure to function as conditions evolve. Plants respond to repeated herbivore pressure by shifting defense location. Instead of uniformly increasing deterrence, they concentrate protection near vulnerable tissues. This targeted strategy conserves resources while maintaining effectiveness. Intelligence prioritizes defense placement rather than magnitude, reflecting refined internal assessment. Plants assess internal oxygen diffusion efficiency and modify tissue porosity accordingly. In dense or waterlogged environments, air channels expand to facilitate gas movement. This adaptation maintains respiration under challenging conditions. Intelligence integrates physical constraints into anatomical design, enabling survival where oxygen is limited. Plants detect internal imbalances in lipid composition and adjust membrane synthesis. Proper membrane fluidity is essential for enzyme function and transport. Temperature shifts and stress alter lipid needs, prompting recalibration. Intelligence maintains cellular functionality by fine-tuning foundational structures rather than surface features. Plants adjust growth symmetry in response to uneven resource distribution. When light or nutrients favor one side, growth compensates to maintain balance. This correction prevents structural instability and maximizes exposure. Intelligence preserves coherence, ensuring that asymmetry does not compromise function. Plants alter internal nitrogen recycling efficiency during scarcity. Older proteins are broken down and reused to support essential processes. This internal scavenging reduces dependence on external input. Intelligence transforms limitation into opportunity through resource recovery rather than depletion. Plants respond to prolonged darkness by reorganizing metabolism rather than halting activity entirely. Energy use becomes conservative, and alternative pathways sustain vital functions. Intelligence adapts to absence rather than waiting passively for return of light. Plants regulate cell division rates based on tissue stress levels. Damaged or strained areas slow division to prevent compounding weakness. Healthy regions continue expansion. Intelligence balances growth with integrity, avoiding fragile overextension. Plants integrate temperature and moisture signals to assess frost risk. Cold alone may not trigger full protective responses unless accompanied by dryness or specific timing cues. Intelligence evaluates compound conditions, reducing unnecessary activation. Plants adjust internal pH buffering capacity under metabolic stress. Enzymatic reactions depend on narrow chemical ranges, requiring constant regulation. Intelligence maintains chemical stability even as activity fluctuates. Plants alter secondary compound profiles as communities change. new neighbors provoke different chemical expressions than long-established ones. Intelligence responds to social context rather than fixed identity. Plants coordinate leaf turnover with nutrient availability. Shedding occurs when resources can be reclaimed efficiently. Intelligence times loss to maximize recovery. Plants regulate vascular diameter in response to flow demand. Increased transport widens conduits, while reduced demand allows narrowing. Intelligence optimizes infrastructure dynamically. Plants adjust internal signaling speed based on urgency. Strong disturbances accelerate transmission, while mild conditions maintain slower pace. Intelligence modulates tempo rather than content alone. Plant intelligence reveals itself as continual calibration. Through sensing gradients, filtering noise, allocating resources, and anticipating change, plants persist within complex environments. Their cognition is distributed, patient, and precise, unfolding through structure and chemistry across time. Plants measure not only light intensity, but also subtle shifts in spectral quality, allowing them to anticipate shading from neighboring vegetation. Far-red light increases under dense canopies, signaling potential competition, which prompts elongation of stems or reorientation of leaves. This early detection preserves photosynthetic efficiency before full shading occurs. Intelligence manifests as foresight expressed through growth patterns, where biochemical perception translates environmental gradients into structural adaptation. The plant essentially reads its surroundings to preempt disadvantage, integrating temporal and spatial information seamlessly. This nuanced awareness demonstrates that plant intelligence relies on interpretation of indirect signals rather than direct confrontation or motion. Plants exposed to periodic drought optimize stomatal behavior to maintain internal water balance. Stomata partially close to prevent excessive transpiration while still allowing gas exchange. Over repeated cycles, these adjustments become fine-tuned, reflecting accumulated experience encoded within tissue responses. Root growth also adapts, extending deeper in search of stable moisture sources. By balancing water conservation with photosynthetic necessity, plants maximize survival without compromising energy acquisition. Intelligence emerges in the integration of physiological and structural strategies, allowing plants to navigate fluctuating hydration environments in a way that resembles adaptive planning, despite the absence of nervous tissue. Plants detect and respond to subtle soil chemistry gradients created by microorganisms, mineral availability, or decaying organic matter. Roots grow selectively toward beneficial compounds while avoiding harmful concentrations, effectively mapping a dynamic underground landscape. This targeted exploration minimizes wasted energy and enhances nutrient uptake. Chemical signals are continuously processed at multiple scales, influencing cellular metabolism and organ-level growth. Intelligence is evident in this capacity to interpret complex, multi-layered information and translate it into spatially precise action. Rather than reacting randomly, the plant demonstrates problem-solving through selective growth informed by its immediate chemical environment. Plants exposed to repetitive herbivory adjust defensive strategies temporally and spatially. Rather than uniformly producing deterrent compounds, the plant concentrates protective molecules on tissues most likely to be attacked. Simultaneously, it may accelerate repair in lightly damaged regions. These responses are calibrated based on frequency and intensity of past damage, creating a dynamic and anticipatory defense pattern. Intelligence is expressed in strategic allocation. The plant invests where it matters most, conserving energy and resources. Over time, these measured responses create an adaptive memory, reflecting environmental experience encoded in chemical and structural adjustments rather than neurological processes. Plants perceive minute variations in atmospheric pressure and humidity, which guide processes like flowering, seed dispersal, and transpiration timing. Sudden changes may indicate storms or drought, prompting physiological adjustments to reduce risk. By integrating these cues with internal hormonal signals, plants optimize survival outcomes. Intelligence emerges as predictive responsiveness. Plants act in anticipation of environmental shifts rather than in reaction. This capacity transforms subtle physical signals into coordinated biochemical and structural changes. The plant's perception of the atmosphere functions as a continuous data stream, allowing it to align growth, reproduction, and defense with external conditions in remarkably sophisticated ways. Plants modulate photosynthetic enzyme composition in response to fluctuating light quality. Rather than simply increasing or decreasing leaf area, chloroplasts reorganize, and pigment ratios shift to capture energy more efficiently under shaded or filtered light. These adjustments preserve energy balance and minimize photo damage, representing an internal fine-tuning process. Intelligence is evident in the plant's capacity to optimize performance through biochemistry, rather than brute force expansion. Over time, repeated exposure conditions leaf physiology for similar scenarios, creating a form of experience-driven adaptation that enhances long-term survival without conscious awareness. Plants adjust root architecture in response to the distribution of soil nutrients over multiple seasons. When mineral patches are localized, roots grow directionally toward high concentration zones while minimizing growth in depleted areas. This dynamic restructuring conserves energy and maximizes nutrient acquisition. Intelligence emerges from spatial evaluation, pattern recognition, and adaptive decision-making, executed through biochemical signals. The plant essentially maps the soil environment internally and responds in a calculated manner. This capacity allows survival in heterogeneous soils where indiscriminate growth would be inefficient, demonstrating that intelligence can be encoded in growth patterns and chemical signaling rather than neural processing. Plants exposed to repeated temperature fluctuations alter membrane composition to preserve fluidity and function. Saturated and unsaturated lipid ratios adjust in response to both sudden and chronic changes, stabilizing cellular processes. Protective proteins accumulate to safeguard enzymatic activity, and metabolic pathways are modulated to reduce energy loss. Intelligence is expressed through anticipatory preparation. Structural and biochemical changes occur in advance of catastrophic damage. By translating environmental experience into persistent internal adjustments, plants demonstrate a form of memory and foresight embedded in molecular architecture, ensuring survival across variable and unpredictable thermal conditions. Plants synchronize reproductive timing with environmental cues such as light, temperature, and humidity, integrating multiple signals to determine optimal seed dispersal periods. Hormonal pathways coordinate floral development, pollen release, and fruit maturation to align with peak pollinator activity or favorable growth conditions. This synchronization maximizes reproductive success while minimizing resource expenditure. Intelligence is present in the integration of multiple temporal and spatial signals, guiding complex developmental decisions. Through these coordinated adjustments, plants demonstrate foresight and strategic planning at the biochemical and structural level, optimizing reproductive outcomes without requiring mobility or conscious awareness. Plants regulate internal energy allocation in response to resource availability and demand. Photosynthetic output is directed preferentially to growing tissues, storage organs, or defensive compound synthesis, depending on immediate and anticipated needs. This dynamic resource budgeting ensures stability under fluctuating environmental conditions. Intelligence emerges from the ability to prioritize, redistribute, and balance internal reserves to maintain growth, reproduction, and defense. By continuously evaluating the state of energy, nutrient, and structural requirements, plants implement a form of decision-making embedded in chemical signaling, demonstrating that adaptive management of internal resources is a key expression of plant intelligence. Plants adapt leaf angle and orientation to optimize light capture throughout the day and across seasons. Movements are gradual and guided by photoreceptors that sense both intensity and spectral composition. Adjustments reduce self-shading, prevent photo damage, and enhance overall photosynthetic efficiency. Intelligence manifests as geometric optimization, integrating environmental information to guide structural change. Over time, repeated exposure reinforces effective orientation strategies, embedding environmental learning into morphology. This process allows the plant to continuously harmonize its form with fluctuating light conditions, illustrating cognition expressed through form and function rather than neural activity. Plants detect airborne chemical signals emitted by nearby plants experiencing stress, allowing anticipatory responses. For example, volatile compounds released from damaged neighbors may trigger increased production of defensive chemicals or growth adjustments in unstressed plants. Intelligence is evident in the interpretation of external chemical cues and the ability to adjust physiology proactively. This form of interplant communication enhances survival and competitive advantage, demonstrating that plant intelligence encompasses not only self-regulation, but also environmental perception extending into community-level interactions. Plants exposed to salinity, stress modify root ion transport and osmotic balance to maintain cellular integrity. Sodium and chloride ions are sequestered into vacuoles or expelled, while compatible solutes accumulate to preserve turgor. Growth patterns shift subtly to prioritize organs less affected by salt exposure. Intelligence is embedded in adaptive chemistry, allowing survival under conditions that would overwhelm unmodified tissues. By translating external chemical stress into precise internal adjustments, plants demonstrate problem-solving that does not require mobility or awareness, but relies on continuous feedback integration. Plants modulate stomatal density over developmental time in response to historical CO2 levels. High atmospheric CO2 may trigger lower density to reduce water loss, while low CO2 promotes increased stomata for effective gas exchange. Intelligence emerges through long-term calibration, allowing plants to anticipate environmental trends and adjust physiology preemptively. These structural adaptations integrate environmental history into growth, reflecting a slow yet highly effective learning process encoded at the tissue and cellular level. Plants adjust flower morphology and timing in response to pollinator availability and competition. Petal orientation, nectar production, and scent emission may shift subtly to attract preferred insects while minimizing energy expenditure. Intelligence is revealed in the coordination of structural, chemical, and temporal traits to maximize reproductive efficiency. By fine-tuning reproductive signals, plants demonstrate strategic optimization within ecological networks, aligning their developmental programs with external rhythms to enhance survival and reproduction. Plants exposed to partial defoliation accelerate leaf replacement selectively, prioritizing photosynthetically valuable tissues. Growth hormones redistribute to compensate for loss, restoring energy capture without overproducing unnecessary leaves. Intelligence is expressed through adaptive reallocation and prioritization, preserving functional efficiency under stress. Over time, repeated exposure shapes tissue responsiveness, embedding environmental experience in developmental programs. Plants exposed to fluctuating nutrient availability refine transporter expression dynamically. Uptake proteins in roots become highly sensitive to localized surges while downregulating in depleted regions. This fine-scale modulation conserves energy and ensures maximum absorption efficiency. Intelligence lies in the spatially precise interpretation of resource distribution, enabling optimized exploitation of transient opportunities. Through repeated cycles, plants develop persistent responsiveness patterns encoded in transporter regulation and root architecture. Plants integrate internal circadian rhythms with external environmental cycles to coordinate metabolism, growth, and defense. Temporal alignment ensures optimal photosynthesis, energy allocation, and stress responsiveness. Intelligence is evident in this synchronization, which allows internal processes to anticipate daily and seasonal variations. By linking internal clocks with environmental cues, plants achieve adaptive timing that enhances efficiency and survival without requiring movement or sensory consciousness. Plants detect soil compaction and adjust root architecture to navigate physical obstacles. Growth direction, branching frequency, and elongation rates shift to exploit less resistant paths while avoiding stress zones. Intelligence operates through mechanical sensing and adaptive restructuring, maintaining access to water and nutrients even in restrictive conditions. The plant effectively maps its underground environment, responding dynamically to physical constraints and translating them into optimized growth patterns. Plants regulate internal pH across tissues in response to metabolic fluctuations and environmental acidity. Enzyme activity, ion transport, and secondary metabolite production are adjusted to maintain chemical stability. Intelligence emerges through continuous homeostatic monitoring, enabling sustained function under variable conditions. By integrating chemical feedback with structural adjustments, plants preserve equilibrium and ensure coordinated operation at the cellular and organ level. Plants adjust canopy density strategically in response to light competition. When shading threatens photosynthetic capacity, leaves may elongate or reposition, while in low competition, lateral expansion dominates. Intelligence expresses itself through growth optimization, balancing light capture, structural integrity, and energy expenditure. Through continuous assessment of environmental feedback, plants modify form preemptively rather than reactively, demonstrating long-term strategic planning embedded in growth patterns. Plants exposed to repeated pathogen presence modify surface chemistry preemptively, increasing antimicrobial compounds or signaling molecules. These changes are localized to vulnerable tissues and timed with anticipated infection cycles. Intelligence operates through predictive defense and spatial prioritization, optimizing protection while conserving resources. The plant integrates environmental signals and internal status to guide subtle, anticipatory responses that enhance survival under persistent microbial threat. Root systems detect moisture gradients across centimeters of soil, expanding toward richer areas while minimizing energy spent in depleted zones. This selective growth pattern ensures efficient nutrient absorption, while dormant zones are temporarily ignored, conserving resources. Environmental history informs this behavior, with repeated hydration cycles creating long-term responsiveness encoded in tissue structure. Feedback mechanisms coordinate cellular expansion, chemical signaling, and enzyme activity to navigate heterogeneous substrates. Intelligence manifests through spatial mapping and dynamic decision-making. Without awareness, the organism translates localized signals into growth patterns that optimize survival, transforming otherwise chaotic underground environments into structured zones of opportunity. Leaf surfaces adjust their orientation in response to subtle changes in light angle and spectral composition. Chlorophyll density, pigment distribution, and venation patterns are recalibrated to maximize energy capture while avoiding photodamage. Circadian rhythms coordinate these movements, integrating daily fluctuations with seasonal expectations. This form of responsiveness extends beyond simple growth. It represents anticipatory alignment, where internal biochemical states harmonize with external conditions. The adjustment is gradual, precise, and layered, reflecting an embedded intelligence that continually interprets environmental cues, optimizes resource intake, and maintains balance between exposure and protection, creating an invisible choreography of survival. Hormonal signals traverse the vascular network to coordinate responses across distant tissues. Nutrient shortages in one region trigger redistribution of carbohydrates and amino acids toward areas under active development, while surplus reserves are sequestered for later use. This dynamic allocation functions as a form of internal logistics, ensuring structural stability and metabolic efficiency. Rather than reacting indiscriminately, feedback loops prioritize essential growth zones, repair pathways, and defense mechanisms simultaneously. Intelligence arises from distributed coordination. Independent units respond in harmony to both local and global signals, generating a cohesive, adaptive outcome without central control. Chemical cues emitted by neighboring organisms influence defensive and growth strategies. Volatile compounds and root exudates indicate stress, predation, or competition, prompting preemptive biochemical adjustments. Receptors detect subtle differences in composition, concentration, and timing, triggering localized or systemic responses. Experience shapes sensitivity, creating persistent responsiveness patterns in tissues exposed repeatedly to such signals. Intelligence emerges from the integration of social and environmental information. The organism modifies allocation, growth, and defense strategies in anticipation of events beyond its immediate boundaries, achieving adaptive outcomes in a complex ecological network without motion or consciousness. Repeated mechanical stress, such as wind or abrasion, gradually alters structural tissue properties. Fibers align along stress axes, cellular walls thicken in vulnerable areas, and flexibility is calibrated to reduce fracture risk. These incremental modifications produce a form of physical memory. Future responses to similar forces occur more efficiently and with reduced energy cost. Intelligence in this context is expressed through anticipatory structural adaptation, where cumulative experience informs material properties and architecture. The organism does not perceive consciously, but continuously transforms mechanical feedback into long-term resilience encoded in anatomy and composition. Exposure to fluctuating temperatures prompts biochemical and structural adjustments to maintain cellular stability. Enzyme activity is modulated. Membrane composition shifts between saturated and unsaturated lipids, and protective proteins accumulate in anticipation of stress. Over repeated cycles, these responses become refined, creating durable tolerance to extremes without compromising growth. Intelligence manifests as predictive acclimation. Physiological systems interpret environmental trends and optimize internal states to prevent damage. By encoding experience into chemical and structural modifications, survival becomes a dynamic negotiation with thermal conditions rather than a passive reaction. Reproductive timing is synchronized with multiple environmental cues, including photoperiod, temperature patterns, and resource availability. Developmental pathways coordinate floral maturation, pollen viability, and seed formation to align with peak pollination or dispersal windows. Hormonal and chemical signals integrate these inputs, balancing energy expenditure with reproductive success. Intelligence arises in temporal optimization. The organism anticipates favorable conditions and adjusts growth, maturation, and investment accordingly. Through this intricate coordination, survival and proliferation are maximized across fluctuating environments, reflecting adaptive planning embedded within structure and biochemistry rather than through sensory perception. Surface chemistry dynamically adapts to microbial and herbivore presence. Defensive compounds, antimicrobial metabolites, and signaling molecules are produced selectively in response to localized threats. Spatial and temporal modulation prevents wasteful overproduction while ensuring vulnerable regions remain protected. Intelligence expresses itself through prioritization, spatial awareness, and chemical feedback loops. By interpreting both past interactions and present stimuli, the organism allocates resources efficiently and maintains resilience against persistent stresses. This chemical strategy allows survival and continued growth even under variable biotic pressures, demonstrating a sophisticated interplay of memory, anticipation, and response without a nervous system. Resource allocation shifts between growth, defense, and storage depending on internal needs and external conditions. Carbohydrates, proteins, and secondary metabolites are redistributed dynamically. Actively developing regions receive priority, stressed areas are reinforced, and surplus is stored for future demands. Feedback mechanisms evaluate energy balance continuously, preventing overextension or depletion. Intelligence is evident in this ongoing triage. The system integrates local signals, metabolic status, and environmental data to optimize outcomes across the organism. Efficiency emerges not from central planning, but from distributed, coordinated responsiveness, allowing survival and adaptation in complex, changing conditions. Vascular networks adjust conductivity and flow rates according to developmental demand. Regions under active growth receive increased transport of water and nutrients, while dormant areas receive reduced flow. These adjustments are mediated by chemical signaling and pressure gradients, creating dynamic internal logistics. Intelligence is embedded in this regulation. Flow is continually monitored, redistributed, and fine-tuned in response to changing needs. Through decentralized coordination, transport efficiency is maximized, waste minimized, and growth and repair processes are synchronized. This continuous self-management enables the organism to thrive in spatially and temporally heterogeneous environments without centralized oversight. Root systems adaptively explore soil based on micro-scale chemical gradients, extending preferentially toward nutrient-rich zones, while avoiding regions high in toxins or competition. Subtle ionic differences and pH variations guide directional growth, while prior experience influences responsiveness, creating a form of memory embedded in tissue structure. Dynamic communication between cells coordinates branching, elongation, and transporter activity to maximize absorption efficiency. Intelligence emerges as distributed problem-solving. Environmental inputs are continuously processed, decisions are enacted at cellular levels, and collective growth patterns generate optimized strategies, enabling survival in complex and unpredictable subterranean landscapes without central control or conscious awareness. Leaf orientation adjusts continuously to optimize light capture while minimizing photo damage. Photoreceptors detect changes in angle, intensity, and spectrum, prompting gradual shifts in laminar position and venation function. Circadian cues integrate with seasonal changes to maintain efficiency across hours, days, and months. Movements are subtle but precise, balancing exposure and protection, energy gain, and water loss. Intelligence expresses itself through anticipatory calibration. Environmental information is translated into structural adjustments, preserving function and stability. The organism interprets complex light patterns holistically, creating a living, responsive surface that maximizes resource acquisition while harmonizing with fluctuating conditions, a slow yet profound choreography. Hormonal networks coordinate internal resource allocation between storage, defense, and active growth. Signals originating in stressed, developing, or dormant regions traverse vascular conduits, triggering redistribution of sugars, amino acids, and secondary metabolites. Feedback loops monitor adequacy and adjust transport continuously, avoiding overinvestment or neglect. Intelligence emerges as decentralized planning. Multiple autonomous tissues contribute to holistic balance, ensuring growth, survival, and repair are optimized simultaneously. These dynamic internal logistics, achieved without neural computation, represent a sophisticated form of adaptive management, allowing the organism to reconcile competing priorities while navigating temporal and spatial resource fluctuations. Chemical communication extends beyond immediate tissues, interpreting cues from neighboring organisms to anticipate potential threats or opportunities. Volatile compounds, root exudates, and airborne metabolites convey information about herbivory, microbial presence, or competition. Receptors decode subtle variations in concentration, timing, and composition, initiating preemptive, defensive, or growth responses. Intelligence is evident in this capacity to integrate external social signals with internal state, producing anticipatory adjustments. Adaptive responses are spatially and temporally refined, demonstrating that survival strategies are informed by both self-assessment and environmental context, creating a distributed, proactive awareness embedded in biochemistry rather than cognition. Mechanical forces sculpt internal structure over time. Repeated wind, rain, or abrasion induces fiber alignment, differential wall thickening, and tissue flexibility adjustments. Incremental modifications reduce fracture risk and enhance stability, creating a persistent memory of stress encoded physically. Intelligence manifests as anticipatory structural adaptation. Experience alters material properties, optimizing resilience for future challenges. Responses are neither reactive nor conscious, but emerge from continuous feedback between mechanical input and developmental processes. Over multiple cycles, these subtle shifts produce durable architecture capable of withstanding recurring disturbance, illustrating that cognition can exist in distributed physical form without neural circuitry. Temperature fluctuations drive progressive biochemical and structural recalibration. Enzyme function, membrane fluidity, and protective protein accumulation are adjusted according to both immediate stress and historical exposure. Over repeated cycles, tolerance becomes reinforced, creating a predictive response to environmental variability. Intelligence is expressed as dynamic acclimation. Chemical and structural systems continuously evaluate conditions and optimize internal states to prevent damage. By embedding experiential data into physiology and tissue properties, survival becomes an adaptive negotiation rather than a passive reaction, demonstrating foresight encoded at the cellular and molecular levels. Reproductive processes synchronize with multiple external cues, including light patterns, thermal cycles, and moisture availability. Developmental timing coordinates flower maturation, pollen viability, and seed formation with peak pollinator activity or optimal dispersal conditions. Hormonal networks integrate these signals, balancing energy expenditure with reproductive success. Intelligence emerges through temporal optimization. Complex environmental information is processed and translated into precise developmental adjustments. Anticipatory alignment enhances reproductive efficiency, ensuring that energy investment coincides with favorable conditions. Survival and proliferation are maximized through coordinated responses across tissues, demonstrating a form of strategic planning embedded in structure and biochemistry rather than reliant on perception or mobility. Surface chemistry adapts dynamically to biological pressures, including microbial colonization and herbivory. Defensive compounds and signaling molecules are deployed selectively in vulnerable regions, while non-essential tissues are spared, conserving resources. Spatial and temporal modulation optimizes protection without compromising growth. Intelligence is expressed through prioritization, feedback integration, and anticipatory defense. Prior exposure informs the timing and location of chemical responses. Repeated encounters reinforce adaptive patterns, embedding environmental memory into physiology. The organism navigates biotic stress with strategic chemical precision, demonstrating distributed cognition, realized through chemical, structural, and enzymatic coordination rather than centralized processing. Internal energy and nutrient allocation shifts according to developmental, environmental, and stress-related demands. Active growth areas receive priority, storage organs accumulate reserves, and defensive processes are upregulated as needed. Continuous monitoring ensures balance between supply and demand, preventing overextension or waste. Intelligence is evident in this dynamic triage system. Resource distribution is guided by real-time feedback and internal assessment rather than fixed programming. Through distributed coordination, efficiency is maximized, allowing the organism to thrive under fluctuating conditions and maintain equilibrium across competing physiological demands, an elegant form of embedded adaptive management. Transport networks within vascular tissues adjust conductivity and flow based on localized and global requirements. Regions undergoing active expansion receive increased hydration and nutrient flow, while dormant or structurally stable areas receive less. Pressure gradients, chemical signals, and local feedback loops coordinate these changes continuously. Intelligence manifests in dynamic self-organization. Distributed units optimize infrastructure to match current and anticipated needs, maintaining functionality and structural integrity. By constantly evaluating demand and rerouting resources, the organism ensures survival in heterogeneous environments, demonstrating that adaptive problem-solving can emerge from decentralized, physically mediated systems rather than cognitive awareness. Chemical signals traveling through vascular tissues regulate responses to localized stress, such as drought or pathogen attack. Hormones move rapidly from affected regions to distant tissues, prompting preemptive adjustments in metabolism, growth, and defense. Feedback loops fine-tune intensity and timing, ensuring energy is not wasted. Intelligence emerges from the integration of signals across scales. Local disturbances inform global adaptation, producing coordinated physiological adjustments. This decentralized system acts as a predictive network, translating environmental cues into structural and biochemical outcomes, demonstrating a subtle form of cognition embedded in chemical communication and responsive architecture rather than centralized neural processing. Microscale light fluctuations trigger pigment rearrangement within photosynthetic organelles, optimizing energy absorption while preventing photodamage. Chloroplasts shift positions, and accessory pigments recalibrate their ratios in response to intensity, spectrum, and angle. Circadian and seasonal cues synchronize these adjustments with broader environmental cycles. Intelligence is expressed through anticipatory optimization. Energy capture is maximized while damage is minimized, achieved without movement or consciousness. Over repeated exposure, these dynamic adjustments are refined, creating persistent patterns that improve efficiency and resilience. Light becomes both a resource and an information channel, shaping structural and chemical responses in a continuous adaptive dialogue with the environment. Mechanical stimuli, from wind to water flow, provoke subtle tissue remodeling. Fibers realign, walls thicken selectively, and elasticity is adjusted according to stress orientation. Incremental changes accumulate, embedding a memory of environmental forces into structural design. Intelligence manifests through anticipatory adaptation. Prior mechanical experience informs future responses, reducing the likelihood of structural failure. Coordination occurs at the cellular and tissue levels without central control, producing emergent resilience. Over time, these adjustments create an architecture that is both flexible and robust, translating continuous feedback from the physical world into durable internal modifications that enhance survival in a dynamic environment. Nutrient pulses influence the timing and direction of root expansion. Areas with transient abundance trigger accelerated growth, while depleted zones are deprioritized. Transporter proteins and enzymatic activity adjust to maximize uptake efficiency. Repeated exposure to fluctuating availability creates persistent responsiveness encoded in root physiology. Intelligence is expressed as strategic exploration. Resources are sought selectively, and energy is allocated according to opportunity and historical patterns. The system does not predict in a conscious sense, but integrates chemical signals and past experience to generate efficient growth behavior. This adaptive allocation allows survival in heterogeneous soils where indiscriminate growth would be wasteful and ineffective. Reproductive structures align development with multiple environmental signals, including temperature trends, photoperiod, moisture, and biotic activity. Timing is critical. Pollen viability, nectar production, and seed maturation must coincide with optimal dispersal and pollinator availability. Hormonal networks integrate these inputs, creating a coordinated developmental program. Intelligence emerges through temporal synchronization. Developmental decisions reflect integrated environmental assessment, maximizing reproductive success. Anticipatory alignment ensures energy investment is efficient and effective, revealing sophisticated strategic optimization encoded in biochemical and structural networks rather than through movement or neural computation. Defense responses adjust spatially and temporally according to past damage and exposure. Secondary metabolites, structural reinforcements, and antimicrobial compounds are concentrated where risk is greatest, while unaffected regions remain energetically economical. Feedback loops monitor intensity and duration of stress, refining response patterns over repeated events. Intelligence is expressed as selective prioritization. Adaptation is targeted rather than uniform, guided by both local stimuli and historical experience. This approach preserves resources while maintaining resilience, demonstrating a form of distributed cognition in which chemical and structural systems coordinate proactively across the organism. Internal energy allocation dynamically balances competing demands from growth, storage, and defense. Carbohydrates, proteins, and metabolites are rerouted to meet immediate and anticipated needs, with priority given to critical tissues. Feedback mechanisms evaluate supply and demand continuously, preventing depletion or inefficiency. Intelligence manifests in this ongoing triage. Resource management is adaptive, distributed, and responsive, integrating internal status with environmental cues. This allows survival and functional continuity in fluctuating conditions, illustrating that sophisticated prioritization and optimization can emerge without central processing, relying instead on networked signaling and feedback. Hydration fluctuations prompt subtle adjustments in tissue permeability, osmotic regulation, and transport activity. Water movement is directed toward regions under high metabolic demand, while surplus is stored or redistributed. Repeated exposure to cycles of scarcity and abundance refines these responses, creating persistent internal efficiency patterns. Intelligence is expressed through anticipatory homeostasis. Physiological systems integrate external conditions with internal status to optimize survival. The organism continuously monitors and responds to hydration gradients, translating complex environmental information into precise structural and chemical modifications without awareness or movement. Airborne chemical cues from neighboring organisms influence anticipatory adjustments in metabolism, growth, and defense. Volatile organic compounds signal herbivory, disease, or competition, prompting preemptive changes in protective chemistry and resource allocation. Sensitivity is refined over repeated exposure, creating persistent responsiveness patterns. Intelligence is expressed in the capacity to integrate external social and environmental information into internal decision-making, shaping growth and defense strategies proactively. Coordination occurs across tissues without central control, producing adaptive outcomes that enhance resilience in a community context, demonstrating cognition embedded in chemical perception and internal regulation. Vascular architecture adapts dynamically to changing transport demands. Conduits expand or constrict in response to flow requirements, while pressure gradients and chemical signals coordinate distribution. Regions of active growth or repair receive prioritized resources, whereas stable or dormant areas maintain minimal allocation. Intelligence is expressed through decentralized optimization. Feedback-driven adjustments maximize efficiency and ensure structural integrity. By continuously monitoring and responding to internal demand, the organism maintains functional balance across tissues and organs. This distributed regulation transforms local and global environmental inputs into coherent, adaptive management of internal networks, a sophisticated, non-conscious problem-solving system. Roots grow toward water and nutrients in the soil by sensing tiny differences in moisture and minerals. Some areas attract more growth, while others are left alone, saving energy. Cells send signals to coordinate this growth, so branches spread efficiently in the right directions. Over time, repeated patterns of water and nutrient availability create a kind of memory inside the roots. Decisions about where to grow are made without a brain, but the result is smart exploration. The system balances effort and reward, showing how life can solve problems quietly through growth and chemical communication across tissues. Leaves slowly turn or tilt to catch the most sunlight during the day. Tiny sensors detect how bright the light is and which direction it comes from. Cells inside adjust the structure of the leaf to gather more light for energy while avoiding damage from too much sun. Patterns of light over time help the organism learn the best angles for efficiency. Movements are subtle but continuous, creating a living surface that constantly adapts to its surroundings. This careful adjustment shows a kind of intelligence in responding to light patterns without needing nerves or a brain. Signals travel through tubes inside the body to move food, water, and other important chemicals to the parts that need them most. Areas that are growing fast get more energy, while quiet areas get less. Feedback from each part tells the system where to send resources next. This is not random. It is balanced and thoughtful, like a silent plan. By constantly checking supply and demand, the organism makes sure nothing is wasted and all parts are supported. This shows intelligence as careful internal organization, where energy and resources are always directed where they are needed. Nearby organisms give off chemicals into the air or soil, and these messages are received and acted on. When one is under attack or stress, the signals warn others to prepare for possible danger. Defensive chemicals or growth changes happen in response, keeping the body safe. Repeated signals make tissues more sensitive over time, so the response is faster next time. This system works without a brain or senses, but creates a network of awareness through chemistry. The ability to act before damage occurs shows intelligence built into communication and response across the whole organism. Wind, rain, or touch gently stress tissues, and over time the structure adjusts to become stronger. Fibers realign and walls thicken in areas that face pressure. These changes make the organism more resilient, so it bends without breaking. Past experience with stress is remembered in the way new tissue grows, creating long-lasting strength. Intelligence appears in this slow adjustment, where form is shaped by experience and repeated conditions. The organism responds carefully, improving stability while conserving energy. Even without movement or nerves, this silent adaptation allows survival in a world full of physical challenges. Energy and nutrients are moved to growing or repairing parts as needed, while less active regions are kept minimal. Surplus is stored for future use, and defenses receive resources if stress occurs. Signals constantly update the system on needs, making adjustments smooth and efficient. This balance prevents waste and ensures survival in changing environments. Intelligence is seen in this careful management, where resources are directed thoughtfully, not randomly. Growth, repair, and defense are coordinated quietly, showing how adaptive organization can happen without a central brain or conscious control. Leaves and other surfaces adjust chemical coatings to protect against insects or disease. Compounds are produced where they are most needed, and unnecessary areas are left alone. Timing and placement are based on signals from past experience and current threats. This careful distribution saves energy while keeping the organism safe. Intelligence shows in prioritizing actions and reacting to patterns over time. Each chemical adjustment is small but meaningful, creating a system that predicts risk and strengthens defenses in the right places. This silent, adaptive behavior demonstrates clever responses without requiring a nervous system. Internal networks change flow rates of water and nutrients depending on need. Tubes widen or narrow, and pressure adjusts so energy reaches the right parts at the right time. Growth, repair, and protection all receive attention when necessary. Feedback ensures balance across the whole organism. Intelligence is expressed in this ongoing adjustment, where supply meets demand efficiently. Actions are coordinated without awareness, yet the system adapts to changes in the environment quickly and effectively. This shows that problem-solving can happen through simple, continuous feedback loops rather than brains or thoughts. Timing of reproduction is carefully matched to the environment. Temperature, daylight, and moisture all influence when flowers, seeds, or fruits develop. Chemicals inside coordinate development, so energy is used at the right moment. This ensures that new life has the best chance to survive and spread. Intelligence is seen in the careful scheduling of these processes, anticipating the environment and adjusting internally. The body reacts to subtle cues and organizes growth in a way that maximizes success, quietly planning for the future without needing awareness or mobility. Cells adjust themselves when nutrients or water are scarce. Some break down older parts to recycle materials, while others slow growth to save energy. Distribution is carefully balanced, so vital areas get what they need first. Over time, the organism becomes efficient at managing shortages, reacting faster each time stress occurs. Intelligence is shown in this thoughtful internal economy, where materials are used wisely, recycled, and prioritized. Survival is maintained not by luck, but by continuous assessment and adjustment that makes the whole system more capable under changing conditions. As our journey through the secret intelligence of plants comes to a close, allow yourself to sink even deeper into relaxation. Feel the steady calm of roots anchored in earth, the quiet confidence of leaves turning toward light, and the timeless patience of growth, unfolding molecule by molecule. Let the idea of this green living network, communicating, adapting, and sustaining life, gently fade into a comforting sense of connection. Your thoughts soften, your muscles loosen, and your breath flows naturally like a slow breeze through a moonlit forest canopy. Rest here, fully supported in this safe and serene space. If you enjoyed this relaxing moment, please consider leaving a like, subscribing, or sharing your thoughts below. Your support is deeply appreciated and helps others find their way to this peaceful space. You can also turn on the notification so you don't miss any of our future journeys together. But for now, let your body relax, release any tension, and allow your mind to drift. Good night and sleep well.